Hello there everyone, welcome back to Ants Authority. Today we will discuss study on the role of anti-insulin protein in longevity of queen ants. In this video, we will discuss about this topic in detail, so let's start the video. On 1st September 2022, a research article about the role of anti-insulin protein in the longevity of queen ants or pseudo-queens was published in Science. Claude Desplan and a few other researchers found that the queens of Harpegnathos saltator produce more insulin for egg production, but ovaries also produce anti-insulin to control the aging-related pathways. These ants have an essential role in reproduction and egg laying. An entire colony depends on them to maintain the population in the nest. In addition, they do not live for only a few weeks or months, as their internal signal transduction pathways support long-term survival by suppressing aging. Reproduction significantly influences longevity because the genes that improve the reproduction rate are also responsible for reducing the lifespan. It is observed that the reproductive ants produce more insulin because this hormone promotes the production of eggs, which is a high metabolic activity and requires plenty of energy. It regulates MAPK signaling pathways involved in the reproduction process and allows the reproductive ants to produce a bulk of eggs. However, this hormone is not favorable for their survival due to its involvement in the pathways related to aging. It also regulates the AKT signaling pathways and shortens the lifespan. The researchers have studied the negative impact of insulin on the AKT signaling pathways, which are known to promote the survival rate and growth responses. Hyperactivation of AKT signaling pathways is related to aging in the muscle cells, so insulin is indirectly responsible for the aging and shorter lifespan. The researchers at New York University conducted a study on Indian jumping ants to learn about the reasons for longevity in queen ants. It was interesting to know the reasons for their longer lifespan when the reproduction process is typically related to the shorter lifespan in other organisms. Claude Desplan, Huayan, Long Din, Alexandra Lipholz, and a few other researchers conducted research on the caste switching behavior of saltator. They compared the expression of genes in the brain, ovary, and fat bodies of worker ants and pseudo-queens that had switched caste in the absence of queens. After comparing gene expression, they observed that insulin production increased in their brain. It correlated with an increase in lipid and vitellogenin, which is suitable for eggs. They found that insulin promotes egg-laying abilities in ants, and several insulin-like growth factor signaling pathways also get activated during reproduction. The anti-insulin protein does not affect the MAPK pathway, but this leads to decreased signaling of the fork-head box or branch in the fat bodies. They inspected the ovaries of gametes and found 4 to 6 egg chambers with developed oocytes, while workers and revertants had 0 to 2 egg chambers with no developed oocytes. They performed RNA sequencing of tissues involved in metabolism and reproduction and found differential expression of genes in the gametes workers and revertants. In addition, they also found differences in the size of fat bodies, as the worker's fat body was white and bigger than gametes, yellow-colored and smaller fat bodies. On 1st September 2022, they published results in the journal Science and shared their observation about the differences in the gene expression and metabolic processes of worker and queen ants. Thanks for watching the video.